Thanks, Jeff, for the introduction. Today, I will be presenting preclinical considerations for cell and gene therapy products, an FDA perspective. This slide gives an overview of my presentation. I will review the diversity of OTAT-regulated products in oncology, describe a preclinical testing program in the 2013 FDA guidance for preclinical assessment of cell and gene therapy products, discuss animal species models consideration in preclinical assessments. I will have an in-depth discussion of safety assessment considerations for genetically modified cellular products, cell-based immunotherapy products, gene therapy products, including oncolytic viruses and microbial vectors, and genome editing and cell-based products incorporating the genome editing technology. I will then discuss early communication with CBER OTAT with a focus on INTERACT. Finally, I provide selected FDA guidance in the topics that are discussed in my talk. As my CMC colleagues Kim and Bo have described, OTAT-regulated products in oncology include ex vivo genetically modified cells, such as CAR-T cells. Non-viral vectors, such as plasmids, are another product class for therapeutic or vaccination use. Replication-deficient viral vectors, such as adenovirus, are on the list and have broad application for generating tumor vaccines or delivering transgenes. Replication-competent viruses can be oncolytic viruses, such as conditionally attenuated adenoviruses, vaccinia, herpes viruses, or such viruses engineered to express transgenes. Microbial vectors used for gene therapy include bacterial vectors such as Salmonella, Listeria, or E. coli, which can be genetically modified to express human tumor antigens or therapeutic proteins or nucleotides. Human genome editing or edited products are those that incorporate genome editing technology in the genome of cells. Tumor vaccines can be vectors engineered to express tumor antigens, peptide-based vaccines, or mRNA-based vaccines. Cell-based vaccines are grouped with cell-based immunotherapy products in the last bullet for this talk. Examples for cell-based immunotherapy products will be provided in the next slide. Examples of cell-based immunotherapy products include CAR-T cells and TCR transgenic T cells that are derived from the concept of tumor infiltration lymphocytes, TILs. These products are designed typically to introduce desired specificities and enhanced activities against the tumors. However, they also introduce risks, which will be discussed further in the following slides. There are emerging class-engineered non-T cells, typically B cells and NK cells, which have been designed to introduce additional mechanisms for immune-based recognition and activation for tumor killing. The immune cells can be derived from technologies of stem cell and induced pluripotent stem cells. Characterization of these cells should be comprehensive depending on manufacturing processes. Cell-based therapeutic vaccines are derived from cells such as dendritic cells, irradiated tumor cells. Cell-based immunotherapy products can be derived from autologous or allogeneic cell sources. This slide shows the overall regulatory pathway that should be followed during the development of a cell gene therapy product with a focus on preclinical phase. According to the regulation FDA 21 CFR Part 312, preclinical data should be submitted in an initial IND for human studies. The middle box on the left highlights the preclinical expectations in an IND submission. The FDA has generated the various guidance documents to communicate with the investigators about the preclinical expectations. The documents published by International Council for Harmonization, ICH, are also important resources for guiding the preclinical assessment. OTAT provides regulatory and scientific input on the preclinical program via informal and formal meetings. Ongoing early communications with OTAT, pharmacology, toxicology staff during product development help to ensure that regulatory expectations related to safety, demonstration of potential activity, and understanding of possible mechanism of action are addressed. The first FDA guidance on preclinical assessment for cell and gene therapy products was published in 2013. It provides preclinical study considerations for objectives and general program design. It provides recommendations for assessment of cell and gene therapy and therapeutic vaccines to support an initial IND submission. It also provides recommendations for additional non-clinical testing to support an eventual BLA. It incorporates a strategy for 3 R's, that is, reduce, refine, replace animal use in animal testing. Importantly, the guidance specifies a science-driven review process to address safety issues in a context that considers both the biology of the product and the intended clinical indication. This approach incorporates the basic toxicology principles in preclinical testing while introducing a product-specific testing strategy. My talk is derived from the principle and the strategy of the guidance. The 2013 guidance specifies preclinical program objectives, which should be addressed to inform the design of first-in-human study. The first objective is to support the rationale for the first-in-human study from preclinical proof of concept POC studies that describe the intended product for its bioactivity and the pharmacologically effective dose range, the durability of these reactions. An important note is that the cell and the gene therapy products are to be administered directly into disease subjects. Therefore, bioactivity of the product shown in appropriate animal models can help in interpreting the expected biological responsiveness in the intended disease population. The next objective is to demonstrate the safety for administration to humans. This involves a comprehensive set of testing of toxicology, biodistribution, and other individual testing to permit identification, characterization, and quantification of potential local and systemic toxicities, their onset, that is acute or delayed, the possibility for resolution of any toxicities, and the effect of product dose level on toxicity findings. The third objective is that preclinical data should inform the clinical trial design in selection of initial safe starting dose level, dose escalation scheme, and the dosing schedule. The adequacy of a dosing plan should also be determined by preclinical data in animal models that address the risk to subject population and the clinical route of administration. Preclinical data should also allow for identification of physiological parameters to inform clinical monitoring to mitigate the risks. Ultimately, a careful analysis will be incorporated in our review process to inform a balance of benefit versus risk profile in the context of the particular clinical indication under study. This slide provides considerations for selection of animal species models. The animal species selected for preclinical assessment should demonstrate a biological response to the investigational cell and gene therapy products based on the expected effectiveness in humans in order to generate data to inform clinical trial design. In the determination of the relevant species, these factors should be considered. That is, compatibility of physiology and anatomy to that of humans, permissiveness, susceptibility to infection by the vectors, and the replication of viral vectors or microbial vectors, and immune tolerance to a human cell therapy product or transgene product expressed by the virus or vector. This would involve use of non-standard test species in contrast to healthy animals that usually represent the standard model test system employed in conduct of the traditional toxicology studies. For cell and gene therapy products, study designs using animal model of disease are frequently modified to incorporate important safety parameters that allow for assessment of potential toxicity. For example, tumor models should be used to assess the safety of the product that is selectively active in tumors. Such data can supplement or possibly be used in lieu of toxicology study in healthy animals. The use of syngeneic tumor-bearing models versus human xenograft models is also depending the goal of testing. For example, the use of syngeneic immunocompetent tumor model enables assessment of immune-related risks, while use of immunodeficient tumor model of human xenograft is not adequate to assess this type of risk. Another example is the use of transgenic animals in testing safety of a cell gene therapy product in a human-related background for recognition and activation process in animals. In some cases, use of companion animals with human-related tumor is beneficial. On the other hand, possible issues with animal model availability and technical feasibility may exist and would warrant further data to justify selecting alternative approaches. Before moving to the discussion of specific aspects of preclinical assessments, I thought it would be helpful to give an idea about preclinical data sources. Sources of data to support an IND consist of GLP-compliant toxicology assessment conducted by a certified testing facility, well-controlled studies conducted in-house, published data in peer-reviewed journals, cross-reference to similar products in previously submitted files to FDA, and detailed clinical data from clinical trials. This slide highlights potential safety concerns for cellular products. Administration of cellular product may be associated with inflammation and immune response in the host. This response may be acute or delayed after administration. There might be delayed adverse events associated with inappropriate cell proliferation or cell differentiation leading to tumor formation or unwanted tissue formation. These cells may spread to undesired normal tissues. Homing of administered allogeneic cells very often causes graft versus host disease in multiple tissues. For genetically modified cellular products, there are additional safety concerns. The major safety concerns are about the toxicity related to gene modification, typically cell-mediated on-target off-tumor activation in the normal tissues, from tissue cross-reactivity to the targeting, and off-target activities. Activation of undesired immune response, typically cytokine release syndrome and immune effector cell-associated neurotoxicity syndrome, are the major adverse events associated with administration of CAR-T or TCR-modified T cells. In addition, vector use in genetic modification can introduce potential risks of genotoxicity and tumorigenicity. In vitro and in vivo data should be provided to address the POC and the safety of a CAR T cell product. The coming 3 slides will provide examples of preclinical data used to assess a CAR T cell product. We will review the in vitro data that demonstrate on-target activation assessed by cytokine release assays and the cytolysis of target tumor cells. Data are also needed to address target specificity. This data may come from studies to characterize antigen-dependent T cell proliferation and protein arrays to screen for potential reactive epitopes in a human protein database. To assess off-target activity, we will review data from expression profile of intended target in the human tissues, in silico analysis to predict the potential off-target sites, and in vitro off-target testing against the cells from various non-target tissue sources. Tissue cross-reactivity should be addressed using test articles with the same targeting molecules that is a single-chain variable fragment on a comprehensive panel of human tissues. In addition, assessment of genomic risks related to novel integrating vectors should be considered. We will assess in vivo data generated in appropriate animal models regarding proof of concept in anti-tumor activity and the survival benefit and the safety assessment for in-life and terminal analysis, cell expansion, and persistence following administration. Data generated by using an analogous T cell product in animal models can be applicable provided that data are available to justify the similarities between the surrogate and the clinical T cell product in assessing anti-tumor or on-target activity and off-target risks. On the other hand, differences in the immune system between animals and humans will be taken into consideration when the outcome is interpreted for safety. We also consider additional supporting data for assessing the rationale and safety for clinical administration, such as initial clinical dosing plan. This data may come from previous clinical experience with similar T-cell products, particularly those T-cell products that incorporates the same CAR as CFV. In this regard, clinical data derived from investigational approved equivalent monoclonal antibody with identical specificity are also supportive in the determination of clinical administration of the CAR T-cell product. The published experience with the same target is also taken into consideration. I'm now switching to genome editing technology considerations, as this technology has been applied to some of the OTAT regulated products in oncology. Current genome editing technologies commonly use site-specific nucleases to introduce a double-stranded DNA break. There are 4 families of engineered nucleases: CRISPR-Cas systems, meganucleases, TALENs, and zinc-finger nucleases. GE components may be delivered by viral vectors, plasmid, mRNA, protein, ribonucleotide proteins in order to process for genome modification of cells ex vivo or in vivo by direct administration of the engineered GE components. My today's discussion for preclinical assessment of genome editing will focus on the ex vivo cellular products that incorporate genome editing. There are unique aspects of genome editing processes. They are involving insertion, deletion, or replace the DNA sequences in the genome, generation of double-stranded breaks by the nucleases, endogenous DNA repair process of the double-stranded breaks by non-homologous end join, NHEJ, or homology directly repair. NHEJ can result in errors of the repaired sequences, while homologous direct repair are less errorsome than NHEJ. Our preclinical considerations for these products are based on the risks of on- and on-target effects, genotoxicity, chromosome translocation, and tumorigenicity in relation to nuclease-cleavage of the cell genome. It is important that nuclease-cleavage is well characterized in order to guide the subsequent assessment. We review the data on characterization of the specific on-target site editing, use sequencing-based methods. We assess the data on characterization of genome editing related off-target sites occurring in the genome using orthogonal approaches, typically in silico prediction and deeper sequencing of the predicted cleavage events, biochemical and cellular approaches to identification of off-target sites, and the whole genome sequencing of the edited DNA samples. We also review data to assess potential risks associated with off-target and undesired on-target effects. Particularly, this data should help assessing the impact on coding genes and functional non-coding sequences, chromosome integrity, cellular physiology, and cell transformation. Now I'm switching to products of gene therapy, oncolytic virus, and microbial vector. The safety concerns for these products vary depending on the types of vector and the viral used, their biodistribution and persistence in non-target tissues and biofluids, typically blood, and the level of viral replication in the body. Inappropriate immune activation by the vector virus transgene is a major safety concern. Vector institutional mutagenesis and oncogenicity would be a concern for certain integrating vectors, such as oncolytic retroviruses. Risks of the delivery procedure would be concerned for certain novel procedures. This slide describes the types of preclinical testing for the product of gene therapy, oncolytic virus, and microbial vector. POC can be assessed using in vitro and animal models of disease to demonstrate the bioactivity for anti-tumor activity and the survival benefit. We encourage assessment of biodistribution starting with POC studies to gain idea for vector virus present in target and non-target tissues. Safety, that is toxicology and BD, can be assessed in healthy models to ensure sufficient study duration to be included. In some cases, safety data can also be obtained in animal models of disease to incorporate POC, BD, toxicity. These hybrid study designs should be considered on products that are selectively activated in the presence of tumors. The coming two slides provide safety study design considerations. The study should allow non-biased design, particularly randomization of study animal groups, mimic the planned clinical scenario as close as possible, and administer clinical vehicle formulation at multiple dose levels of the test article, in which the intended clinical starting dose level and the maximum dose levels should be bracketed. The clinical product should be used in the safety study. Justification should be provided for the use of a surrogate to the clinical product. The study should include adequate numbers of animals per group, multiple sacrifice time points, and sufficient study duration. Comprehensive safety assessments for in-life parameters, immunogenicity, macroscopic analysis, and histopathology are expected. Non-terminal assessment, for example, imaging, may be incorporated to assist in safety analysis. Biodistribution assessment is an important component of safety evaluation. The coming three slides highlight the considerations based on the BD reflection paper by FDA and international regulators, and the FDA guidance for industry as a reference. As a definition of BD, assessment of the pharmacokinetic aspects of these products is performed by BD determination. BD is defined as the distribution, persistence, and the clearance of the product in vivo from the site of administration to target and non-target tissues, including biofluids. BD assessment typically include analytical methods for the collected tissues and biofluid samples to detect the vector virus genomes, but may also include methods to detect the expression product. BD can be assessed as a separate study or as a component of a pharmacology or toxicology study. BD should be assessed in a vehicle control group and a group of animals that received the maximum dose level in the toxicology study. Several sacrifice intervals should be included to determine the profile. Samples of blood and a core list of tissues should be collected. That is, injection site, gonads, brain, liver, kidney, lung, hot, and spring to assess systemic distribution of the administered product. Depending on the product type and the tropism, transgene characteristics, and the route of administration, other tissues may be added. For example, draining lymph nodes, bladder, urine. It is important to avoid the potential for cross-contamination among different tissue samples in sample collection. BD assay method is to be sensitive and quantitative to detect the product sequences. A qPCR assay is the current standard method. Additional product-specific testing may be performed to address concerns related to tissue-specific toxicity, genotoxicity, tumorigenicity, or others as applicable. The testing may be incorporated in the definitive safety study. This slide describes conduct of safety studies. Each toxicology study submitted should be performed in compliance with good laboratory practice per 21 CFR Part 58. For non-GLP studies conducted in-house, oversight of the conduct of the study and the resulting final study report by an independent QA unit or person per 21 CFR Part 58.35 is recommended. I echo my CMC colleague Kim that we encourage communication with OTAT at the early stage of the product development, and INTERACT is previously known as pre-pre-IND interactions. CBER implemented the INTERACT program since October 2018 and generated a link to INTERACT meeting process and designated a contact in the SBIA website to provide a definitive structure. The purpose of INTERACT is to provide a mechanism for early communication which may benefit the investigator via non-binding, informal scientific discussion with CBER review disciplines, that is CMC, PharmTox, and Clinical, on a product development program for novel and complex investigational product. For processed INTERACT, you initiate the contact. Typically, when you have a generated preliminary data, POC, and some safety, but are not yet ready to conduct definitive preclinical safety studies. You provide a concise briefing package, approximately 50 pages, with key issues for consideration clearly identified. Representatives from the OTAT review disciplines review each request meeting package and determine if it is appropriate for an INTERACT meeting based on the allowed scope. The pharmacology toxicology information in the briefing package contains the following: comprehensive summary of all completed in vitro and vivo studies that come from POC studies, preliminary safety studies, and the related references. Description of the preclinical development plan consisting of completed and planned studies intended to support the rationale and safety of product administration in humans. Specific questions you would like to discuss regarding your submission. However, the design for definitive safety studies are discussed in detail in the pre-IND meeting. Pre-IND meeting is a formal meeting to communicate the initial development plan with the 3 review disciplines at CBER in order to facilitate an eventual IND submission. For preclinical testing program, discussion will be on comprehensive summary of the data from the completed studies and a plan to conduct definitive safety studies. In summary, comprehensive product characterization is key to understanding product risk. The preclinical testing program may need to be adapted to the specific cell gene therapy product and level of perceived risk. New in vitro and in vivo test models should be considered as the science and technology advances. The 3 R's should be applied to preclinical testing programs. Communication with FDA at early stages of product development may be beneficial. This slide provides a list of selected FDA guidance. I have discussed the 2013 clinical guidance and the 2017 draft guidance regarding pre-IND meetings and the 2020 long-term follow-up guidance in reference to BD assessment. The 2015 clinical guidance describes the approaches to preclinical information for the clinical study design and should help guiding a translation from preclinical to clinical studies. This slide gives the contact information, including my email, if you have further questions about the presentation. This concludes my presentation. Thank you for listening. Now I'm open for questions.
Oh, wow. Thanks, Dr. Huang. So, I mean, your talk highlighted the fact that, complex biological therapies, cells, viruses, gene editing, also entail more questionably relevant models for toxicology and also, proof of concept models. I mean, if you think it's hard for sponsors and sometimes in CDER to figure out what's a relevant species, I mean, it's got to be impossible sometimes to figure out what's a relevant species for you guys, particularly if it's a novel viral vector. I mean, I'm like, do you guys in these guidance documents, such as Bo mentioned the Newcastle virus for some, oncolytic viruses, do you guys reference, okay, for a Newcastle virus, this is a relevant species? I assume if you're using influenza, maybe a ferret is a relevant species. And with these animal models that are relevant for certain species, are they, if they're like different than normal GLP, CROs are used to handling? Well, who does those, or do you do those in house, or are there CROs out there that use these relatively not frequently used models to do GLP studies?
Thanks, Jeff, for your questions. I think you have asked several questions, which are key questions. And first, I think if I missed some of your questions, just interrupt me. First, I think your question is about how we determine the relevant animal models. I think we will need some data. For example, if you were thinking about a virus, oncolytic virus, Newcastle virus, or some adenovirus, for example. What we want to see whether the animal you choose, typically if it's a healthy animal, whether these animals are supporting the position to the virus, which is very important for adenovirus, because adenovirus is a human virus, and usually rodents, it's not permissive to rodents. It's also permissive to some other, not permissive to some of the non-rodents. So that we need some data to show that if you are thinking about adenovirus as an oncolytic virus, and we need some testing for typically the animal we consider like a hare, hamster, or cotton rat. Those animals are relatively permissive. I think cotton rats serum hamster. I consider may not be the typical rodents, but they are permissive to adenovirus. So those are animals that we consider are relevant to test for testing of safety for those types of oncology virus. And Newcastle virus, as you mentioned, it's a typical. I think it has much broader tropism, if you put that way, for the selection of the animal species. So as long as sponsors provide the evidence that the animal they choose that has the permission that supported the replication of adenovirus, or the other aspect is would support the assessment of immunogenicity, which sometimes could be difficult to do in a single animal species. Then the sponsor needs to provide some justification how they address those safety issues related to viral replication as well as immunogenicity. That's one question. The other question you asked whether the regular GLP facility would have the capability to conduct the GLP toxicology study using those animals. And then I think we recognize these issues. So then we implement some aspects for studies conducted in-house, but we do need some quality assurance from the studies conducted in-house based on CFR Part 58. Does that
yeah, unfortunately, it's a little hard to hear you. So if you could speak a little slower and get a little closer to your mic for the next question, that might be helpful. Yeah, no, it's helpful. And so you guys, I mean, clearly complex biological products that are alive are hard. Science is hard, and figuring out how to do the regulatory quality control, clinical trial attributes for things that are alive is even harder. So I get it, and I know you guys have the INTERACT program, which I applaud completely. And I know it sounds like you clearly are trying to be flexible, and that if for some reason you had to use a ferret, maybe there is no CRO that's able to do that. I'm going to go to some of the more granular regulatory questions here, and one of them seems relatively simple. Can you just expound and opine more on therapeutic cancer vaccine?
Sure. I think as I took some examples for the cancer vaccines that could be involving multiple products for gene therapy, for cell-based immunotherapy, or some typical things like mRNA-based, peptide-based, those vaccines. I think we all consider this as a type of gene therapy, and then we would also ask for proof of concept information, particularly whether those products have the ability to have the therapeutic effect is a tumor vaccine. And also, I think we also will depend on, I think most of the novel products that we also will ask for some safety information. However, if the product has been used in the clinical, or you have some similar, for example, peptide vaccine, you have similar peptide vaccine products, those safety data or proof of concept data may be able to cross-reference for your intended product.
Great, thanks. It's still a little bit hard to understand you, not language-wise, but just because it's muffled, and I think it has something to do with the audio. Try to follow Jeff Kelly's instructions on the presentation chat there while I read this next question to you. So people are not quite sure they understand the INTERACT program, and I think I get it. There's a CATS program that helps people understand CMC, technology, product issues, and then the INTERACT program is more for these questions that you just discussed. But maybe you could go over that and just clarify that a little more, the INTERACT versus the CATS and versus a pre-IND program.
Thanks, Jeff. As I think my colleague Tim already elucidated that PET is for CMC is technology. Interact is designed is for a process for handling requests for the for early communication. For I would say it's more for early communication for some novel and complex products involving complex manufacturing and novel subjects, and also involving complex device development. So those are more product-wise. You already have the sponsor, already have an idea what the product will look like. They really like to have some early communication with the FDA to get some idea how they can proceed with the characterization for preclinical aspects and also for safety. For which would lead to safety, but it's really this type of product-wise discussion for interact.
Great, thanks. Yeah, no, I know you don't. I know you don't accept or approve every pre-interact IND submission. So hopefully those guidance documents, where possible, help sponsors understand what would be relevant and applicable models for various types of products. Unfortunately, I'm gonna go early. Thank you so much for participating, and I'm gonna move earlier to our next speaker. But thank you so much, Ying. You've been really helpful, and it's clearly a big topic, and I applaud both yours and Mercedes and the rest of CBER's